Psychology, Psychology 109, and this is the first e-lecture. This is going to be Chapter 1. The title of Chapter 1 is Biopsychology as a Neuroscience. So what is biopsychology? Well, the important thing to remember here is that when a word starts with, you might have multiple terms in a word, as in the case of biopsychology. So you have biology, and then you have psychology. The important thing to know here is that whatever comes first is the primary component of this study, field of study. So in this case, biopsychology, biology is the primary, psychology is the secondary. We'll learn many terms as we go through the chapter where psychology will come first, and then maybe the physiology will be latter. But for the most part, in this chapter, what we're going to discuss is what is biopsychology and what do biopsychologists do. Your author, Dr. Pinnell, goes over 4 major themes in the book. One of those is thinking creatively about biopsychology. Creativity is thinking outside the box, and it is also evidence-based. Okay, there's also clinical implications. So oftentimes what we know about biopsychology came from individuals who have had brain ailments or damage to the brain, and we're able to study those individuals and learn more about ourselves. And new knowledge leads to new treatments is definitely the case. Number 3 is the evolutionary perspective, the consideration of environmental pressures on human evolution. And often biopsychologists use a comparative approach, which means that you might compare different species in your study. And neuroplasticity, the brain is plastic, not static, and this is a very good thing. What this means is that the brain is malleable. The brain can change. In the last few years, we have discovered that the neurons can actually regenerate. Believe it or not, when I was in graduate school, you can look at the textbooks, and there was no such thing as regeneration of neurons anywhere in the brain, and the thinking has definitely changed nowadays. Okay, so the e-lecture topics for week number 1, you'll notice that in the PowerPoint presentation, there are slides that are kind of slipped in through the material, and what I'm going to request of you is that you answer each one of these, and then you submit your items when you do your initial post for your discussion thread. Okay, so number 1 says describe the neurological deficits of Jimmy G, and I have a little 5 there in parentheses. That means that one is worth 5 points. E-lecture topics for each week will be worth 20 points. Okay, so you'll want to respond to each one. Also, when you respond in your discussion thread, say at the top e-lecture topics number 1, and then your reply. In fact, it would be great if you even put number 1, describe the neurological deficits of Jimmy G, and then answer. Number 2, Jimmy G's condition was a product of what? Okay, some of them will be very short, some of them will be more lengthy. So the point totals are what you're going to focus on. If it's worth 5 points, spend a little bit more time on it. If it's worth 1 point, it might only be 1 or 2 words to answer that item. Okay. So what is biopsychology? The scientific study of the biology of behavior. Okay, remember what I mentioned earlier. So biology comes first, and then the behavior or the psychology comes second. Psychology is the scientific study of behavior, okay? And it also goes by lots of other terms. You can use psychobiology, you can use behavioral neuroscience. Many people believe that pretty much started with the Organization of Behavior, which was a book written by D.O. Hebb in 1949, I believe, so late 40s there, and that was the birth of biopsychology. Very interesting that we're able to go over a topic such as this today, or even a science that we're really involved with that is so new. Much of what we know, especially when we're talking about the cognitive neurosciences, most of that information is coming out of the 70s, 80s. There was nothing known before that time frame. So here we go. Hebb proposed that psychological phenomena might be produced by brain activity. So Hebb's work helped discredit the notion that psychological functions were too complex to be derived from physiological activities. This, you have to remember that the old school, the Descartes, the dualism, the mind and body separation, that kind of thing, was all part of it. But we now know that through mental deficits, observable behavior is definitely changed. So we alter the brain, and you can alter behavior. So biopsychology utilizes the knowledge and tools of other disciplines of neuroscience. And each discipline studies a different aspect of the nervous system. Let's look at a few of them. Okay? So we have neuroanatomy, which is the structure of the nervous system, not unlike anatomy that many of you probably took in high school or maybe in college as well. The only thing here is that we're focusing on the nervous system, the central nervous system, the autonomic nervous system. So we're referring to this as neuroanatomy. The same with neurochemistry, the chemical basis of neural activity. And we'll be talking a lot about that when we look into neurons and how they communicate. Neuroendocrinology, the interactions between the nervous system and the endocrine system. And I want to pause right here just for a minute, because this is what I think is really important, and one of the key reasons why I'm doing an e-lecture on this is so that you can hear some of these words being pronounced. I don't think there's anything harder than trying to learn terminology for the first time. Everything's foreign to you, learning the terminology from the first time, and you don't know how to pronounce the words. And we're going to be talking about a lot of very, very long words in this science. So neuroendocrinology, one of those, and just being able to say the word, it will make you more comfortable with the word, and you'll be able to memorize its definition. How about neuropathology, which is the nervous system disorders? Neuropharmacology, or the effects of drugs on neural activity. And then finally, neurophysiology, which are functions and activities of the nervous system. So again, not unlike anatomy, which dealt with the structure of the body, neuroanatomy, the structure of the nervous system. Now you have neurophysiology, so it's the physiological aspects or activities of the nervous system. Biopsychological research, human and non-human subjects. I've had the opportunity to work with both in research. I worked in a physio lab where we studied rat species, and we did condition place preference studies. We injected the rats with CNS stimulants or CNS depressants and identified hypermotor activity, and we also dealt with many experimental drugs that we're going through. And this is what we're talking about, a drug that is well before clinical trials, where you would actually work with that drug on a human. So those would be non-human subjects, primarily rats in this kind of research. And then humans. Humans are not going to be nearly as invasive. I perform bio and neurofeedback. I help people deal with anxiety and stress and that kind of thing, and chronic pain. And that's going to be more along the lines of clinical neuropsych. Experiments and non-experiments, definitely in the physiological lab setting, we're dealing with experiments where a lot of things can be controlled, but there's also the case where you might be working with one individual in a case study, and you're not able to control. You didn't randomly select that individual. You didn't randomly place them into groups, because you only have one person. And then there's pure and applied research. Pure research is again going to be coming out of a physiological lab, where it's very hard to make that jump from the rat species directly to the human. It's basically designed for pure research, aside from if you were doing veterinarian research. But you're trying to find out the neural basis, trying to isolate maybe a cluster of neurons, or even down to one neuron, and how it actually works. Yeah, that's much different than applied research, where you're trying to help somebody with their ailments. And in the case of, again, clinical neuropsychology, the individual might have short-term memory loss or deficit with depth perception. And in this case, you're doing applied research to help them with their day-to-day life. So many questions can be about the biology behavior are addressed using human subjects. However, much can be learned from studying the brains of other species at all, like I mentioned with the rat species. Species differences are often more quantitative than they are qualitative. What that means is that a rat brain is going to be much, much smaller than a human brain, but the neurochemicals, the neurochemistry, the neurophysiology is very much the same. So quantity difference is very large, but qualitatively it's very similar. So then why use non-humans? Well, they have simpler brains. You can use a comparative approach, which means across species, and there are fewer ethical restrictions. Although please make sure to understand that coming from a physiology lab, these animals are very well taken care of. There's a veterinarian always on site. The room, the environment, the conditions are very controlled and regulated, and so there are ethical standards. There's probably just as many ethical standards for non-human subjects as there is for human. And then why use humans? Well, humans, they can follow instructions, they make subjective reports, and they are often cheaper to work with because you don't have the whole physio lab where they're actually living there 24 hours a day. So, and being able to ask them, how do you feel during this experiment, or are you becoming anxious or something like that, is something very important that a human can tell you, whereas animal subjects would not be able to. Okay, let's look at a little bit deeper into experiments and non-experiments. We're not going to focus a lot on this. That's more in the realm of research methodology. But when you're working with an experiment, you manipulate variables. Okay? And in non-experiments or quasi-experimental studies, that's when you might not be able to manipulate variables. Again, I mentioned to you the case study. You bring in one individual who is an alcoholic, and then you're trying to work with that person and work with increasing their situation, better their life, that kind of thing. You can't grab 20 people and then make 10 of those people alcoholics and the other 10 not, and then do your experiment. That is highly unethical, of course, and you're not going to be able to do that. So both of these have their place. Primarily, the experiments are going to be done in a physiological psychology lab of some sort, and your non-experiments are more in your applied setting. Placing into groups. So you have between subject designs and you have within subject designs. What that means is in a between subject design, all participants are different. Okay? In a within subject design, they are the same person. So the key within subject design that you might be familiar with would be a pre-test, post-test. In a pre-test, post-test, the individual is taking both tests, so you're comparing the same individual. And in between group designs, that is not the case. They're different people. So the difference between the conditions is the independent variable. That is what you manipulate. You might be in a physiological setting, you might be manipulating the amount of drug that you give a subject. Okay? The effect of the independent variable, or the effect of you giving that drug, would be the dependent variable, and that's what you count. In the case, what I mentioned a little while ago would be hypermotor activity. Does a CNS stimulant increase hypermotor activity? Does a CNS depressant depress or reduce hypermotor activity? And then a confounded variable is a variable that comes into your study and can affect the dependent variable in a negative way. It can actually affect it in a positive way too, but the idea here is that what you manipulated, which was the independent variable, was not the only reason why the dependent variable changed. It could have also been because of this confound variable. So you try to isolate confounded variables as much as you possibly can. Much harder to deal with human subjects than with animal subjects. Quasi-experimental studies, again, studies of groups of subjects exposed to conditions in the real world. So again, we're looking at real-world scenarios. They're not real experiments because, like I mentioned, you can't randomly place them into groups. You can't randomly select them from the population. There are certain things that you cannot control for. You bring in an individual in a case study and you're looking at this individual for depth perception or something, and they're also very anxious and very depressed, and those might be pre-existing conditions to their current condition, and there's not much that you can do about that. That is who that person is. So case studies focus on a single individual like Jimmy G, and you have a couple e-lecture topic questions in regard to that individual, and he's in your chapter 1. They are usually more in depth than other approaches, but they may not be generalizable. What that means is that these individuals working in a physio lab, they make sure that they control everything. They control the temperature and the food cycle and the lights in the room and everything like that. But that's not real world. That's not, that kind of control can't happen in the real world. So oftentimes these studies are less generalizable than if you were doing it in the real world, as would be the case in a case study, possibly. So through experiments, they definitely look at testable hypotheses, and then generalizability is the degree to which results can be applied to other cases or real world. We talked about that. So oftentimes what you'll notice here is that what initially starts in a physio lab with all of your control measures and so on, and your initial hypothesis, you can test a lot of those hypotheses, knowing that they might not be readily generalizable. But then after lots and lots of hypothesis testing, they can work their way out into a more applied setting. And here again, peer research conducted for the purpose of acquiring knowledge, applied research intended to bring about some direct benefit to humankind, and we talked about examples for both of those. Okay. Now we're on to divisions of biopsychology. So there's 6 major divisions, and let's go into more detail right here. Let's start with physiological psychology. Really the basis here is the neuromechanisms of behavior, and they work with controlled experiments. They're working in a laboratory setting. They're primarily doing peer research. They're testing hypotheses, all of these terms that we just talked about. Okay? Now you're able to take those terms and apply them to an actual division of biopsychology. So that would be physiological psychology. Psychopharmacology, control experiments of the effects of drugs on brain and behavior. So again, this is pharmacology. You would go to, let's say you go to Target and you visit with a pharmacist. The pharmacist is an individual who distributes medicine, but that medicine is actually studied and created by a pharmacologist. In this case, these are pharmacologists also, but they work with only brain-altering drugs, only drugs that pass the blood-brain barrier and have effect on the brain. Then you have neuropsychology, psychological effects of brain damage in humans. Usually, again, a clinical emphasis. So I've mentioned to you clinical neuropsychology a couple times. That's what we're talking about here. How about psychophysiology? Now here's a perfect example. In the first item here, we said physiological psychology. So again, the most important part of that would be physiology. Here, it's psychophysiology. So the most important part here is the psychology aspect and then the physiology. And so this would be individuals that might do biofeedback, neurofeedback, and that kind of thing. And then cognitive neuroscience, okay, which is our most recent division of biopsychology, and looking at the neural basis of cognition, why it's been such a division or a newer division and one of most interest now is because of technology. The functional brain imaging, as that technology becomes more and more advanced, we're able to advance cognitive neuroscience. So that makes perfect sense. It's very, very based upon technology. This slide just gives you an idea of a functional brain image. Here, let me draw this for you a little bit. So the blue area right here, that's going to be the prefrontal cortex. So that is the front of the brain. Where you're looking at the colors here, that's going to be at the back of the brain. And so here we notice that a flash of light was brought into the eyes of this subject, and the activity, the neural activity, is back here, which is in the occipital lobe, which is where we find the primary visual cortex. So it would be perfectly plausible for the light energy coming in to be identified by the brain and then cause these areas to become very active. And then lastly, comparative psychology. And what we're looking at here is comparing different species to understand genetics or adaptiveness of behavior. Can be done in the laboratory, can also be done ethologically, which means out in the real world. In this case, most of the time when we talked about real world, it was with humans, but we can also study the way that turtles move in the desert based upon the sun, and we can put them in an environment where it was very, very similar to their actual environment, and then study how they track the sun. Here's a chart, table 1.2, and this goes over each division of biopsychology that we just talked about. Okay, another theme that your author uses is converging operations, or using multiple approaches to address a single question. In the neurosciences, it is highly unlikely that one individual is going to treat a neurological disorder. Okay? Let's say that we take the instance of somebody with a head injury. That individual, depending on the severity of the head injury, they might have to have an operation of some sort. So the neurosurgeon is going to be involved with that. They're probably also going to be looked at by a neurologist. So that neurologist will look at the brainwave patterns and making sure that the individual can still concentrate or sleep or whatever that might be. Then we know that there's going to be chemicals involved as well. There's going to be medications involved. Depending again on the severity of the head injury, they might want to slow down the brainwave activity, and so there's going to be psychopharmacology involved. There's going to be functional brain images done, so there's going to be some cognitive neuroscience there, especially if there's issues with short or long-term memory loss. We can also think about clinical neuropsychology, where the individual is looking at the entire person and how this ailment has affected their way of life. Okay? So you see how there's many different divisions of biopsychology that converge together and solve one problem. Here's an example that Dr. Pinnell comes up with. It talks about Korsakoff syndrome and how Korsakoff syndrome is mostly is a condition with severe memory loss. They didn't know why this severe memory loss was happening. Then they found out that it was primarily seen in alcoholics. So then they said, oh, well, it must be alcohol that's causing it. But in actuality, Korsakoff's is sometimes seen in malnourished persons who have little or no alcohol, and so they've been able to actually track it back to a thiamine deficiency. And it's still due to the alcohol, but what has happened in a severe alcoholic is that they've replaced their regular diet with alcohol, and so they aren't receiving the nutrients that they need to survive. And this slide, again, kind of went ahead on this one, but again, Korsakoff syndrome is the result of thiamine deficiency, but the damage is accelerated by alcohol. Scientific inference, the empirical method that biopsychologists use to study the unobservable. So scientists measure what they can observe and use these measures as a basis for inferring what they can observe. You have to remember that they can't, they're trying to identify, what is the problem with this individual's depth perception. And then they trace it back to, oh, well, they had a stroke in this area of the brain, and those neurons are now not functioning, and that must be the result, or that must be the reason for this. So something that might not be observable directly can still be studied through scientific inference. Okay, and then we have critical thinking again, the ability to evaluate scientific claims by identifying potential omissions or weaknesses in the evidence. Again, evidence-based. We want to make sure that there's the data that supports our decisions. And then finally, well, a couple more here, but here's another one on your e-lecture topic for week number 1. So here you have a case. Case 1. Delgado claims that a charging bull can be tamed by means of stimulation of its caudate nucleus. And this is another little call-out section in your chapter 1. So the question is discuss alternative explanations for the taming of the charging bull. Okay, and it's worth 5 points. That means that you have to elaborate a little bit on that question. It's not going to be a single term answer. Second, what can we learn from this scenario? And that again is worth 5 points. Again, for each one of these items, please post them right at the very beginning of your week 1 discussion thread. And that's how they will be graded. You will have 20 points per week devoted to these e-lecture topics. And then lastly, there was the case of Moniz, where he won a Nobel Prize for the prefrontal lobotomy. And let's see, inadequate postoperative evaluation in human patients, often by the physician, undesirable side effects such as amorality, lack of foresight. So let me give you a little basis here, is that for the prefrontal lobotomy, they initially discovered that it would help some of these individuals with some of the psychological ailments that they were dealing with, and they did a couple of these surgeries. Basically, you look at 20,000 or so of these surgeries that were done based upon very, very few people. So that would be like providing a drug to 20,000 people that was only given to one person in a case study. So you can see how that was not the best case scenario there. And nevertheless, even though some of these people were helped with their extreme illnesses, the side effects were also very severe in some cases. Emotional unresponsiveness, epilepsy, urinary incontinence, and so on. Okay, and so I wanted to end with the Moniz here because that is going to be your discussion thread. That will be the topic of discussion thread for week number 1. So please look at that in your Blackboard shell and work on that homework. And that will be it for Chapter 1 e-lecture. Thank you.